Um, Outside of the brain and the spinal cord, outside of the nervous system, what kind of cells would eat germs and cellular debris?
White blood cells.
The what?
White blood cells?
The white blood cells, exactly. Okay? So within the nervous system, these microglia do a similar job. So when nerve cells die or there's injury to the brain, those cells will self-destruct. The lysosomes will burst open and create cellular debris. And like amoeba, and like white blood cells of certain types, they will eat the germs and cellular debris. Okay?
Professor, the white blood cells eat the germs, and then what do they do with it?
Um,
Does it digest?
Then they often die because they eat the germs, and then they are collected in lymph nodes and other lymphatic tissues like tonsils, the appendix, those places. Okay. We will discuss that when we talk about the circulatory system. Okay. There is a big difference between the extracellular environment within the nervous system compared to the extracellular environment everywhere else in the body. Certain things can pass into the fluids surrounding the nervous system. There is a fluid that circulates around the brain and down and around the spinal cord and then back up through the spinal cord, the little holes in the spinal cord called the central canal. That fluid circulates back up. And then there are cavities in the brain called ventricles that the fluid circulates through. That fluid has a very different extracellular composition of sodium, potassium, chloride, and anions. All right, the high concentration of sodium outside in the extracellular fluid, whatever that number was, that concentration has to be maintained, and so on. Okay, and so in this case, here we have a type of cell that's within that different. What ensures that the environment around nerve cells is kept the way that it should be is a concept called the blood-brain barrier that you might have heard of. That means that certain chemicals don't cross and get into the fluids of the brain, even though that same chemical might get into the extracellular fluids everywhere else. All right. In addition to the microglia, there are ependymal cells. That help create, let me say, they're found in the choroid plexus and ventricles. there are Schwann cells that form, all right, let me shorten this. They myelinate the processes, mainly axons, especially axons. Of neurons of the peripheral nervous system called PNS. That's what we're going to discuss next. Organization of the nervous system. I will, explain all of this in a minute. Then there are oligodendrocytes. These myelinate the processes especially axons. Of neurons of the central nervous system. Which we will discuss shortly. Okay, when we first began our study of the nervous system, we looked at the neuron, the way that the neuron is constructed. We said that the neuron has numerous short receiving structures called dendrites, but each neuron has only one, usually long, sending structure called an axon, okay? Is the way by which neurons of mammals and higher vertebrates are able to send an impulse even faster is by a process called saltatory conduction that involves myelinated axons. Okay, let me explain that. The myelin sheaths are formed by Schwann cells if this is a neuron outside of the brain or the spinal cord. That is, those neurons are part of what we will learn is called the peripheral nervous system, okay? On the other hand, it's oligodendrocytes that form the myelin sheaths around, usually axons of neurons of the central nervous system. The central nervous system includes all neurons that are completely within the brain or spinal cord. What that means is that the whole neuron, its soma, or nerve cell body, its dendrites, and its axons have to be completely within the brain or the spinal cord in order to belong to the central nervous system. And those neurons will have their axons myelinated by oligodendrocytes, okay? Neurons that have any part of itself outside of the brain or the spinal cord will belong to the peripheral nervous system, both of which we're gonna talk about in a few minutes. And so their myelin sheaths will be made by Schwann cells. Okay, now, so similar to the way that an electric cord has insulation on the outside of it. It's the wire inside that's conducting the electricity. Well, likewise, it's the axon itself that's conducting the electrochemical impulse. But it can have insulation in the form of these myelin sheaths. Now, what the myelin sheaths do is they don't so much protect the current in there, but what they will do is allow the action potential to spread down the axon even faster than usual. Okay, so let me explain that. There is a rapid type of transport called saltatory conduction. Saltatory conduction refers to the rapid spread of an action potential that occurs by